This is the final OCR A-Level Biology video on Unit 5. It is on muscles. Muscles cannot elongate without an antagonist. Muscles are arranged in opposing pairs so that when one contracts, the other elongates. The antagonist is sometimes elastic recoil or hydrostatic pressure within a chamber. So it's not always an actual muscle, it can just be the natural elastic recoil of muscles. This table, the reason I'm writing in random columns each is because I originally did it down here and it was in the wrong order for a photo that I put on later, so sorry, just going to have to deal with it. So first of all, smooth muscle. So smooth muscle is made of individual cells, tapered at both ends, so they're spindle shaped. At rest, each cell is 500 micrometers long and 5 micrometers wide. Each cell contains a nucleus and bundles of actin and myosin. They contract slowly and regularly. It does not tire quickly and is controlled by the autonomic nervous system. Involuntary muscle is found within the walls of the tubular structures such as the digestive system and blood vessels. The muscle is usually arranged in longitudinal and circular layers that oppose each other. So the main things with the involuntary smooth muscle, obviously the clue's in the name with involuntary, it's going to be controlled by your autonomic nervous system. I'll show you a picture comparing all three types of muscles after. But then we've got cardiac muscle, and that's the muscular part of the heart. Individual cells form long fibres which branch to form cross bridges between the fibres, and these cross bridges help ensure electrical signals spread evenly over the walls of the chamber, and they also help ensure that the muscle contraction is a squeezing action rather than a one-dimensional contraction action, because obviously we want to squeeze the blood out of these chambers within the heart. These cells are joined by intercalated discs, which are specialised cell surface membranes fused to produce gap junctions that allow free diffusion of ions between the cells. Action potentials can pass easily and quickly along cardiac muscle fibres because of these gap junctions caused by intercalated discs. They contract and relax continuously throughout the life and contract powerfully and does not fatigue easily. So obviously we've got these Purkinje fibres within the heart, all the areas. Heart muscle is myogenic, however it's controlled by the SAN. Finally, we've got this voluntary skeletal or striated muscle. Contraction of skeletal muscles causes a movement of the skeleton by bending or straightening the joint. Muscles are arranged in antagonistic pairs, one contracts while the other elongates. Muscle cells form fibres about 100 micrometers in diameter. Each fibre is multinucleate and is surrounded by a membrane called the sarcomere. Muscle cell cytoplasm is called sarcoplasm. It contains many mitochondria and an extensive sarcoplasmic reticulum. Contents of fibres are arranged in a number of myofibrils, which are contractile elements divided into a chain of sarcomeres containing actin and myosin contractile units. The banding pattern on actin and myosin gives voluntary muscle a striated appearance. And these are your three types of muscle fibre. I realised that when I put this on the video, this image is not going to be detailed enough for you. Just look at it in your textbooks. It doesn't take a few seconds to have a little look at that. So now let's look at the neuromuscular junction, and it's pretty much just a synapse. Nothing really changes here. So skeletal muscle is under voluntary control. Contractions are stimulated by the somatic nervous system. The junction between nervous system and muscle is called the neuromuscular junction, and it's basically a synapse, but it's between the nerve and the muscle fibre itself. So what we've got here, I've literally took this from my other video. This is a axon tip. Previously we would have called it the presynaptic bulb, but here we call it the axon tip. It's got exactly the same process. It's got these calcium ions. We've got acetylcholine within our vesicles. Once the action potential comes down, calcium channels open, acetylcholine containing these vesicles fuse with the ends of the membrane, and it released into the sarcomere, opening sodium channels and causing depolarization, so that this acetylcholine attaches to your receptors, acetylcholine receptors. You get this wave of depolarization produced when we've got sodium moving into our cell, so into the fibre. So sodium moves in here as the acetylcholine receptors open the sodium channels. Sodium then moves in here. We get a wave of depolarization, and the wave of depolarization then moves down these transverse tubules, which are basically surrounded by positive ions, so the negative action potential can depolarize into the transverse tubules and pass between different muscle fibres. Have a little look at that yourself. I understand it. I just went through it really quickly, obviously because mostly this revision is for me, but I do enjoy teaching other people at the same time. Let's now look at this. So skeletal muscle. If you didn't understand what I was talking about, this makes it much clearer. So we've got muscle fibre containing lots of myofibrils together, and each little sort of section here of cells we call a sarcomere. And if we look at, so these two here are basically represented again here. So we've got an I band, a H zone, M line, Z line, or Z line if you watch the American versions of these videos, which I have. It's very painful to watch Z lines. I've started to say it myself. So let's just speak through these bits. So we've got this thin filament actin and this thick filament myosin. So myosin is contained within our A band, which is not on the diagram. So I'm going to draw myosin in black here. So myosin is contained in this area, and each one of these is a myosin filament in the chain, okay? And we call that bit our A band, and that is our dark band, okay? And it will be the same over here. Okay, so we've got myosin contained in this A band, and that's why it appears dark. In the centre, as it points out here, we've got this H zone. So I'll draw two yellow lines around our H zone here. This is what I'm talking about when I'm talking about H zone, this zone here. And this contains only myosin. To make it a bit clearer for you, right, so we've got actin and myosin. Myosin in these black lines are these black lines. Actin is bound to the Z lines, Z lines, however you pronounce it. It's not an oral exam. I've watched so many videos that I now say Z lines because Americans make the videos. Anyway, so actin's bound to your Z lines and it crosses into this A band. So this A band in the centre, one way to remember A band, you can say all. A for all, like, so in case you don't know what I'm saying. A for all, so it contains both actin and myosin within it. I'm quite in my element here because I do A-level PE, which might make some of you scientists frown, or it might make some other A-level PE people happy. Because basically, if we're this desperate to be watching videos this late, we're probably doing A-level PE. Anyway, we've got the H zone in the centre, and the H zone, as you can see, does not contain any actin filaments. So that's why it's a different zone. It's the only zone that doesn't contain actin. I'll do that in yellow. Okay, so our H zone is there in the centre, and a way to remember H zone, H for halfway, so it's in the middle. H zone is halfway. I actually did think of these myself, which is quite proud of. Then we've got this I band, which contains only actin filaments, and let's remember that actin will be going over across both sarcomeres. Of course, I'm just focusing on this sarcomere this time, but actin obviously is going to go to this next sarcomere, And because actin is our light filament, these are our light bands. So I bands are light bands. I can't actually remember my analogy for I. I did make one, but that's on my PE notes. So I bands contain only actin. So what happens during contraction? Our Z lines are pulled inwards towards the centre of the sarcomere. Actin filaments are pulled along the myosin, which stays in the same place. Therefore your A band always remains the same size because it contains the myosin, and myosin never moves. The A band stays the same size during contraction. Then the actin filaments are pulled in, therefore Z lines move in towards the centre of the sarcomere. These actin filaments cross over the myosin filaments within the H zone, so the H zone basically disappears because the actin passes over and it creates that dark band, not the lighter band of the H zone. Your I bands also decrease in size as the Z lines are being pulled towards the centre of the sarcomere, both sides. So if this has been pulled towards there, and it's also been pulled towards that centre of the sarcomere, so the I bands decrease because actin is moving in towards the centre. I bands decrease, Z lines move towards the centre of the sarcomere, H zones decrease, but A bands stay the same. Okay, that is all you really need to know on that. Let's have a look at actin and myosin. So myosin has 2 heads per molecule and bind to actin. We'll look at the sliding filament hypothesis next. And we've got actin, which is 2 subunits twisted around each other. We've got actin, tropomyosin, and this globular protein called troponin. And obviously actin has got to have a receptor for tropomyosin, a receptor for troponin, and also a receptor for calcium. So we get this tropomyosin complex with actin and troponin all working together. And calcium. Okay, so that is all you really need to know about the structure of these muscle fibres. I'm just letting it right out in case there's anything else. Definitely not. Okay, so finally, to end Unit 5, to finish off the whole of... So this is to finish off for Paper 1 next Monday for any of my loyal subscribers watching this year. This is the last video, and this is the last topic in sliding filament hypothesis. And once again, hopefully I'll be good at teaching this because I do PE. And it crosses over. So... hopefully I don't dilute it too much down, but I think it's pretty much the same in both subjects. So sliding filament hypothesis. We've got an action potential. It passes along the sarcolemma and down the transverse tubules. We could call those T tubules. Remember, they absorb that depolarization and take it to the next muscle fibre. The action potential is carried to the sarcoplasmic reticulum, and I've put SR in this, and this sarcoplasmic reticulum stores calcium ions. Calcium ions are then released into the sarcoplasm. Calcium ions then bind to troponin, and this binding to troponin causes it to alter its shape, so it undergoes a conformational change, and it pulls the tropomyosin aside to expose the binding sites on the actin. Once those binding sites are exposed, this myosin head, which naturally has ADP and P attached to it in its resting state, the P is sort of released or expended to allow the myosin head to attach to the binding site on the actin. Then we've just got this ADP. The ADP is then expended for the power stroke, so it pulls along the myosin fibre, so myosin heads attached to it. It pulls along the actin fibre, so it's sort of like, so if you've got a rope attached to a brick, okay, imagine this brick to be your Z line and your hands to be your myosin heads. So what happens, you've got this rope, which is your actin filament, you bind to it, and to bind to it you've grabbed onto it. So you grab onto the rope and that forms these cross bridges. Then you grab on it, ADP is expended, and that's the power stroke, and that's when you pull the rope, you pull that brick towards you, so that Z line moves towards the centre of the sarcomere. You're the myosin, by the way, so your hands are your myosin heads. You're pulling in the rope across your body, and therefore your actin filament is going to move down. Your Z line, which is your brick, is going to move towards the centre. I really hope that's a good analogy. I just thought of it in my head. Then we've got ATP. So obviously you know ATPase hydrolyzes ATP, and to break this cross bridge that we've formed, to allow more cross bridges to form, ATPase hydrolyzes ATP, releases that ATP energy into ADP plus P, and that energy allows the cross bridge to break. Once the cross bridge is broken, we can attach to another binding site, and we can, so basically we let go of the rope with our hands, and that's because the ATP's broken that bond, and we grab onto the rope again if calcium is present, and then we do the power stroke by using ADP, and then we let go again. And each time we grab, pull it and let go, we've pulled the actin, which is our rope, and the Z line, which is our brick, closer to the centre of the sarcomere. So finally, the role of ATP in maintaining its ATP supply. So obviously we've only got about 1 to 2 seconds worth of ATP for contraction. So 1 to 2 seconds worth of contraction with the ATP readily available in muscles. So we've got to constantly regenerate ATP, and that is done through aerobic respiration, and that is assisted by this Bohr effect, because obviously the Bohr effect causes more oxygen to dissociate at the muscles because of the high presence of carbon dioxide. So aerobic respiration is faster in muscles, it's more efficient in muscles. Then we've got anaerobic respiration. This is good for about 2 to 3 minutes until lactic acid starts to accumulate too high, and then we have to stop using that anaerobic system, because as we know, lactic acid will dissociate into hydrogen ions and lactate, and these hydrogen ions cause that conformational change in the shape of the protein that cause more CO2 to bind to it, and less oxygen can be carried on our oxygen molecules. So, and then we get all sorts of fatigue and effect of lactic acid, which you don't need to really know about in biology. Then we've got ATP-PC, which is only used in muscles, I believe. Not something the book goes into too much detail on. Something you go into too much detail in PE. We use the creatine phosphatidyltransferase enzyme to reform phosphocreatine stores, and the breaking of phosphocreatine can supply energy to generate ATP, but this lasts for about 2 to 4 seconds of contraction. That's what it says in my biology book. In PE I'm told it's about 8 to 10 seconds. I don't think the number really matters. Just remember it's a really short amount of time. We're talking seconds here it lasts for. We're breaking down phosphocreatine. We don't get any byproducts from that, and we can resynthesize a lot of ATP, and that's for really powerful contractions. Such as like a shot put throw would use ATP-PC. It's just one short, sharp movement, produces a lot of ATP, which is hydrolyzed really quickly. Okay, so that is the whole of 5.5 in 3 videos. Probably the biggest topic of the whole course. Thank you for watching.